Back with another video. It's Tuesday, another short day in the lab this case, because I only have to prepare some stuff for the experiments of the coming days. So let's do that. Inoculated some colonies. And for the rest of the day I'm just following some lectures, and I also prepared the experiment for the frogs. Wednesday, a whole day of following lectures, of course. Let's go. It's going to be a long evening again. And finishing off with some lectures. The cells are clearly separated from the growth media after centrifuging the grown colonies I inoculated on Tuesday. The 6 tubes here are coming from one inoculated colony. And since I inoculated 6 different strains, I had to do this step and the following step for each of the 6 strains. In the following step, I discarded the growth media of all 6 tubes, so only the cell pellet remained. I resuspended the cell pellet in a specific buffer, and in this way I brought all cells resuspended into one tube. I wrapped this tube with aluminum foil to prevent photodegradation of my protein of interest. Okay, let me quickly explain what I'm doing this week. This week I have 6 different bacteria containing 6 different plasmids, and thus 6 different genes coding for 6 different antibiotics. And I'm going to check each bacteria if they actually produce that antibiotic, that protein. So that's actually everything I'm doing this week. It's going to take 4 days in total. Now I'm already at day 3. I grew the bacteria. Then yesterday someone else induced them, so the proteins were actually produced by the bacteria. And now I'm lysing them with the sonication. So they break open, all the proteins go into solution. Then I will put that protein solution on a Western blot to check if my antibiotics are present or not. But for now, sonication, breaking cells open physically. Alright, 10 minutes have passed. Let's switch the tubes. And the final one is running. Alright, next step is to centrifuge this to separate cell material from the protein solution. This time I discarded the pellet containing cell material you can see at the bottom of the tube, so I obtained a clear solution containing my proteins, as already done for this tube. Right, I didn't film a lot because I don't have a lot of time, but I prepared an SDS-PAGE shell with all my 6 protein solutions, 2 of each. So that's running now, and now I have to prepare the media for the Western blot. Okay, the gels have run, and now one I'm destaining with Combat C and the other one is for the Western blot. I'm removing the delicate SDS-PAGE shell from its plastic enclosure. By the way, if you want to know more details about this technique, I have another video explaining this in detail, link in description. Here I removed the water I used to detach the gel and added the Comasy blue stain, which will eventually allow me to visualize the protein bands. Making some media for the final step of the Western blot. And this is the result of the Western blot. This is now the first antibody solution, which will bind the antibiotic proteins which have a his tag, which I will incubate overnight now. So here's one gel and here's the other. Alright guys, 6:30 PM. I wasn't able to film everything because it was the first time ever we did a Western blot. We still had to figure out some stuff and I had to run pretty fast. And now it's time to go home. And ending the day with lectures of laboratory animal science. Happy Friday! I'm in Ghent. I took the train, it's about 1:30 PM because I have a practical course on like in vitro gut microbiome systems, but now I have to find my bike because I don't remember where I put it and there are lots of them. Found it. Quick push workout. And this beautiful Saturday is spent writing on my literature study of my thesis. And Sunday, of course, again going hard at it. It's 8:30 PM. I'm done writing for my thesis, at least for today. And now it's time to edit this video and post it in hopefully a few hours. Let's do it.